Hey guys, nice to have you back here. Today I'll be diving straight to codons. So what are codons? Just as the name sounds, they are simply codes for specific amino acid, or what are called stop signals during protein synthesis. So, three nucleotides that represent an amino acid or a stop codon on an mRNA makes a codon. As simple as that. The four nucleotide bases remain the same: guanine, uracil, cytosine, and adenine. If you do the math, there are 64 possible codons made up of combinations of these four nucleotide bases. Now, codons are made reference to on RNAs, mRNA to be specific, and mRNAs are made through the process of transcription. The mRNA copies the code from the segment of interest on the DNA for the desired protein to be made. So, for example, if the DNA segment being copied is this here, the mRNA segment or copy would be AUG corresponding to the above. This would be GCU UGC, just based on how the nucleotide bases pair. The codons are then translated to proteins with the help of the tRNA and ribosomal RNAs. The tRNA carries the corresponding anticodons and uses this to bring the desired amino acid. So what are some points to note about codons? The first is that the start codon is always AUG, which codes for methionine. An amino acid. This would mean that most proteins would have the first amino acid to be methionine. The second point is that stop codons, UAA, UAG, and UGA, are not part of the amino acid sequence. Also, they do not code for any amino acid. Hence, how the ribosomes know to stop protein synthesis. The third point I have here is redundancy. Multiple codons can code for the same amino acid. As I've said before, there are 64 possible codons and 20 amino acids. This would mean that multiple codons can code for the same amino acid. Now imagine while sending a message you made a typographical error. This can lead to different kinds of catastrophe depending on who or what the message was intended for. This kind of errors go on in the DNA sequence as well, and it's called mutations. And this can affect codons directly. Mutations can be due to a change in a single nucleotide, and that's called point mutations. An example of point mutations is silent mutation. Here, a single nucleotide is changed or substituted for, but it, however, does not have an effect as the two codons both code for the same amino acid. Another point mutation is the missense mutation. Here, the change in the nucleotide base does have an effect, where glutamic acid is substituted for by valine, and this leads to a common disease called sickle cell disease. Another type of point mutation leads to conversion of a codon to a stop codon, and this can be found in cystic fibrosis. And this type of mutation is a nonsense mutation. There's substitution of C for U. Another type of mutation is one that happens with Tay-Sachs disease. It's called frame shift mutation. Here, there's insertion of a 4-base pair that leads to a shift in the reading frame of the mRNA fragment, altering every other codon from the mutation. In insertion deletion mutation, there's either an increase of amino acid sequence, as seen in Huntington's disease, or a decrease in amount of amino acids, and results in symptoms of cystic fibrosis. In Huntington's disease, the codons that code for the amino acid glutamine increase from about 35 to 36, while in cystic fibrosis, the amino acid phenylalanine is deleted. This concludes my short video on codons. If you found this video helpful, don't forget to like, subscribe, and share it with others. If you have questions or topics you'd like us to cover, drop a comment below. Thanks for watching and see you in the next video.